In the world of early drug discovery, you hear the phrase fit for purpose all the time. But what does it actually mean? A free pass for less rigor? Or is it something much more strategic and intelligent? Let's break it down. One of the biggest mistakes we see in discovery analytics is applying late-stage development level expectations to early-stage discovery decisions. The goals are completely different. Discovery analytics isn't driven by regulatory compliance. It's driven by making the right scientific decisions fast. At this early stage, analytics exists to support medicinal chemistry. The goal is to confirm a compound's identity, get a good estimate of its purity, and spot any obvious red flags or liabilities. You're not trying to build a data package for a regulatory submission. You're trying to enable your chemists to make smart choices about what to synthesize next. So, fit for purpose simply means the data is sufficient to answer the scientific question you're asking right now. Is it perfect now? Is it exhaustive? Absolutely not. But is it sufficient to move the program forward responsibly without getting bogged down? Yes. This brings us to precision versus accuracy. In discovery, relative precision is often way more important than absolute accuracy. For example, if your LC method can reliably tell the difference between a compound that's 85% pure and one that is 95% pure, that's probably good enough to make critical structure-activity relationship decisions. You don't need the validated, hyper-precise methods required for a phase 3 clinical trial at this point. The real metric you should care about is decision confidence. Ask yourself this: does the analytical data allow the chemistry team to correctly interpret trends in potency or other properties? If the answer is yes, then your method might already be perfectly fit for purpose. This is why, for rapid screening, relative area percent from a chromatogram is often totally acceptable. You might not need to perform complex corrected purity calculations or response factor adjustments, unless you have a specific reason to believe an impurity is messing with your biological results. Keep it simple and fast. The danger of not embracing this mindset is over. When you apply phase 3 validation standards to early discovery compounds, you grind everything to a halt. It chews up valuable resources, slows down your project's velocity, and creates totally unnecessary analytical bottlenecks that frustrate everyone. And the cost of slowing down your SAR cycles is huge. In the fast-paced world of drug discovery, turnaround time can literally shape which compounds get prioritized for the next round. An analytical method that takes too long can unintentionally distort the entire direction of your research program. Speed really does matter. Now, this doesn't mean anything goes. Fit for purpose is not a synonym for careless. You absolutely still need clear peak identity. You need reasonable chromatographic resolution, so you're not looking at a jumbled mess. You need your results to be repeatable within practical limits, and you need to be aware of any obvious degradation happening to your compounds. So when is a method not fit for purpose? It's when analytical risk starts to compromise your scientific interpretation. If co-eluting peaks are hiding critical impurities, if degradation products are being completely ignored, or if you can't explain why your potency data is jumping all over the place, then your method is no longer sufficient. Think of it as a phase 0 versus a phase 3 mindset. Discovery operates in that creative, fast-moving phase 0 world. Development operates under the intense scrutiny of regulatory bodies. Confusing these two environments is a recipe for disaster. You either slow down discovery to a crawl, or you weaken your preparation for successful development later on. The key is knowing when to tighten the screws. As a program matures and gets more seriously, when you're re-preparing toxicology batches or your IND submission timeline is approaching that S, when your analytical rigor should progressively increase, but not before it's needed. So, let's wrap up with a clear definition. Fit for purpose analytics in discovery means generating data that is reliable enough to support medicinal chemistry decisions, without slowing down the engine of scientific progress. Ultimately, early discovery analytics isn't about chasing perfection. It's about strategic sufficiency. You need just enough rigor to protect your scientific interpretations, and just enough speed to protect innovation. In our next episode, we'll dive into how to design LCMS strategies that deliver that perfect balance of speed and structural confidence. Thanks for watching, and be sure to follow for more.